All right, welcome back, all of you who are on the call for the fourth day of Thirsting for Theoretical Biology 2021. In this session, I guess for the last three sessions, we've been talking about processes and machinery inside a single cell. And today we will begin zooming out a bit and leading us on these exciting dynamics are going to be Kinneret Keren and Erez Brown from Technion. So this process of morphogenesis, as all of you know, is pivotal and vital in our understanding of biological systems. And what Kinneret and Erez are attempting to do is to put this very interesting dynamics of biological systems into the language of physics, and more specifically that of complex dynamical systems and so on. So there should be a lot of exciting things for us to learn about here. So the first talk is going to be given by Kinneret, who is a professor at the Technion. Her training has been in the condensed matter statistical mechanics, dynamical systems school of physics. And now she's doing these very beautiful experiments on Hydra. And we're going to hear about that from Kinneret. Kinneret, please, the stage is yours.
Okay, so thank you very much for the invitation to talk in this online meeting, and thank you all for tuning in. So as Sashi said, I will be talking about the physics of morphogenesis, focusing on our work on Hydra regeneration, and I'll tell you a lot more about Hydra, which is a small organism which you can cut into a small piece, and then it will undergo whole body regeneration and form a new animal, and we're interested in how this process happens. Okay. Okay, so my lab has been working on mechanical aspects of biological self-organization and pattern formation. We did a lot of work at the single cell level, looking at live moving cells and trying to reconstitute artificial cells that can emulate the cytoskeletal dynamics that we see in cells in a more simplified system. But today I'll talk about our work on Hydra morphogenesis, which is asking at the multicellular level how these self-organization and pattern formation happens. And this is joint work with Professor Erez Brown, who's my colleague in the physics department and who will be giving the second talk today. So the plan for today is, in my talk, I'll first introduce Hydra and explain to you why this is an excellent model system to look at basic questions regarding morphogenesis. And I'll focus on our work on the cytoskeletal dynamics, on the actin dynamics and the influence of mechanics on the process of Hydra regeneration. And then Erez in the second talk will give you a broad overview of physical approaches to morphogenesis and talk about his work on applying external electric fields on regenerating Hydra and showing how you can influence this process and even reverse the process of morphogenesis. Okay, so the process of morphogenesis is basically the emergence of pattern in a living animal. So here you can see an example of embryonic development of a human embryo. So you start with a fertilized egg, which is spherical and round, and then over time it grows into this very elaborate and intricate functional 3D complicated structure. And this is one of the most beautiful examples of pattern formation in nature. And one of the most striking features of this process is what's called canalization. Basically, if you look at this process, the development gives you a very stereotypical and consistent outcome, despite very large variation, both in the system itself and in its environment. So when you look at a baby, it will develop and have 2 hands, 2 eyes and nose, a very stereotypical pattern. And it's really remarkable to understand how this is able to happen in spite of all this variation in the organism and its environment. And one of the fundamental questions, therefore, in developmental biology is how these robust patterns emerge. Another phenomena that allows you to appreciate this remarkable canalization is the process of regeneration. So we know that when an adult suffers some injury or trauma, in many cases it's able to recover this damage or injury, and this is what's called regeneration. And all animals have, to some degree, the ability to regenerate. How much varies between different systems. So, for example, if we take a salamandra and we chop off its limb, it's able to regrow a full limb, including the bone, the nerve, the skin, all these, the tissue, all these complex complex parts of this elaborate tissue, and generate a functional limb within a few weeks. And what's remarkable about that is that it's able to do that from essentially arbitrary initial conditions. If you cut in the shoulder, it will generate the whole limb. If you cut in the wrist, it will only regenerate the digits. In humans, we don't have such elaborate regeneration properties, but you still know if you break a bone, it's able to regrow or heal. Other organisms, such as the liver, are also able to regenerate. And then there are some animals that take this ability to regenerate to the extreme and exhibit what's known as full body regeneration. And this is, here you can see an example of this worm called planaria, and Hydra, which I'll talk about today, also exhibits this. And here, if you cut off the tail of the animal, it's not only that the animal is able to regrow the tail, but the tail is able to regrow the whole animal here within a couple of weeks. So if we think about this process of morphogenesis and the robustness of this process of morphogenesis, regeneration brings a very interesting example of this. So unlike development, where we start with a relatively stereotypical fertilized egg and we undergo through a stereotypical developmental process, in regeneration, we actually start from very variable initial conditions, yet the system is able to form this same final pattern, which is the adult organisms. And the mechanisms involved in development and regeneration are shared. And again, this highlights the main question of how robust patterns can emerge from these variable initial conditions. So if we look at the process of morphogenesis, it's a very complicated process, and it involves an integrated interplay between various biochemical processes, such as various signaling pathways and various morphogens that are involved in patterning the tissue, with mechanical processes that ultimately define the three-dimensional shape of the developing embryo. And what makes this a very complicated process is that there are feedbacks at all levels of organization between these two processes. Now, if you think of this from a physics point of view, what we have here is a many-body, strongly interacting system, very far from equilibrium, yet somehow it self-organizes across scales to form these very robust patterns. And as we know from physics, when we have such a complicated multi-level, multi-scale problem, while it's very insightful to learn about the molecular details involved, you need a coarse-grained effective approach to understand, to approach it from the other direction. So to develop such an approach, what you need is to identify some coarse-grained variable and try to develop a predictive framework to understand the dynamics. And this is the approach we're taking to looking at morphogenesis, trying to take a view that is not at the molecular details, but a coarse-grained view, and trying to see what we can understand about this process and about the physics and mechanics of this process from this point of view. Okay, so the plan for today is first I'll introduce the model system we use to address this question. So it's obviously a very complicated question, which is why it's useful to try to take the simplest model system you have. So and Hydra in this sense has a relatively simple body plan and is a relatively it's not a simple system by any means, but it's a simplified model system that allows us to ask fundamental questions on the process of morphogenesis. So I'll first introduce Hydra as a model system. Then I'll talk about our work on looking at the cytoskeletal organization and specifically on the nematic organization and the defects in this in the nematic order of the actin fibers in regenerating hydra. And I'll show you that during the regeneration process, these topological defects actually can function as organization centers for the process of morphogenesis. This work was recently published last year. And in the second part, I'll talk about newer unpublished work where we focus on the polarity of the regenerating hydra and look at how the memory of polarity and the new body axis is established in the regenerating hydra. And again, showing that the mechanics of this mechanics of the cytoskeletal organization have an important role in this process. And then I'll summarize and conclude, highlighting the role of mechanics and the role of the actin fiber organization as a coarse-grained field that is relevant for this process of morphogenesis, suggesting that the nematic organization of the actin fiber can be thought as a mechanical morphogen in analogy to the chemical morphogen we're familiar from the Turing pattern. Okay, so let me first introduce you to this small creature called Hydra. So this is a small predatory animal. It's a part of the Cnidarian family, which is the same family as jellyfish and coral, which branched off the animal tree quite early on. All these animals are predators. They have stinging cells that they use to for prey. These are distributed in the tentacle, and then they have a mouth and a foot. So basically, we have a uniaxial body plan with a single axis. It has about 100,000 cells from about 10 different cell types. It has a distributed neuronal system, which is actually not essential for morphogenesis. And it gets its name from the mythological Hydra, which would regenerate the head if its head was cut because of its remarkable regeneration properties. So if you take a Hydra and you cut it in half, then the head will regenerate the head will regenerate the foot will regenerate the head. More dramatically, you can cut a small piece of tissue, so a few percent of the whole animal from the mid region of the animal, or you can completely dissociate the animal into individual cells, reaggregate them, and these will form a holospheroid within a relatively short time, and within a couple of days, will reform a full Hydra. So we have here the process of morphogenesis, the generation of a multicellular animal from very flexible initial conditions. It turns out that in Hydra, this process does not require growth. So you can entirely stop cell division and you can get this morphogenesis process. At the end of the day, you will get a smaller Hydra. So it's the same cells, but they reorganize and differentiate to generate the pattern of a mature Hydra. This process happens in solution and the whole process takes only a few days. So it's accessible for imaging and manipulation, which is very useful for experiments. So this is how this process looks like. What we did here, we took a small piece of, we cut a small piece with just a scalpel, a small piece of tissue from the middle part of the animal. And what you're seeing in the movie is after a few hours, it closes into a spheroid, and then it regenerates over the course of a couple of days. So the time here is in hours. And you can see this small piece of tissue start to elongate. You can see here the body axis is defined. You can see the tentacles emerging. You can see the mouth defining. And after 2 days, you'll have a small Hydra. Normally, Hydra reproduce by budding. So the typical way they reproduce is that a small Hydra will grow from the midsection of the adult Hydra, forming this bud. They can also undergo sexual reproduction. This typically happens upon stress. And in general, if you look at adult Hydra, it's actually undergoing continuous tissue replacement. So the entire mid-body of the animal is full of stem cells, and these stem cells undergo continuous divisions and move towards the extremity of the animal. So all this region, which is where you can cut and get regeneration, is continuously dividing. And because of this continuous tissue replacement, actually, there's no evidence for aging in Hydra. So it's also an interesting model system to look at this aspect. Okay, a little more about the structure of the Hydra. So as I said, it has a single axis from foot to head. The shape of the body is basically a hollow tube. And this hollow tube is made of a bilayer of cells. So here you can see this hollow tube. Inside, it's basically the digestive region, the gastric tract. It's filled with fluid. And all the cells reside in this tubular region around this cavity. And these cells are basically divided into two layers. There's an outer layer called the ectoderm and an inner layer called the endoderm. And in between, there's an extracellular matrix. And then there's additional cells, additional stem cell line, the interstitial cells, which will develop into nerve cells and gland cells and all the specialized and the stinging cells, all the specialized cells, which are kind of within these two layers of the ectoderm and the endoderm. And in fact, you can make Hydra with only the ectoderm and the endoderm, and these will still undergo regeneration. Another important aspect of the structure, so these two layers of cells, which are kind of the outer and the inner layer of this animal, also double as muscle cells, and they are supracellular actin structures, which I'll show you in the next slides, which are lining the basal side of the ectoderm along the body axis of the animal, and in the endoderm in a circumferential direction. So these muscle fibers are made of actin-myosin, like the muscles in our body, and these are the main force generators inside our bodies. And here you can see the organization of these supracellular fibers in a mature Hydra. So here you're seeing a transgenic Hydra with GFP life act that labels the actin fibers in the ectodermal cells. And you can see this beautiful array of fibers that basically spans the entire animal from head to foot. Here, just for scale, you can see the scale of individual cells. So these are really, you can look at them, you don't see the cell boundaries. All these actin fibers are obviously within the cells, but they span this mechanically, they're this supracellular structure spanning the whole body. So this is the fibers in the outer layer, which are aligned with the body axis. In the inner endoderm layer, you can see that these fibers form a circumferential array of parallel fibers, and these muscle fibers are the main force-generating entity in Hydra, allowing all the dynamics of the shape that you saw in the movie. So what makes Hydra such a nice model system for looking at morphogenesis? So on the one hand, it's a multicellular behaving animal. You can see here, this is a movie of a Hydra preying on a small shrimp that has tentacles. It stings the little shrimp and it will bring it to the mouth and digest it. So it's a multicellular behaving animal. On the other hand, it has a simple body plan. It has these remarkable regeneration abilities. So it's a very flexible experimental system. You can vary the initial conditions for morphogenesis. You can manipulate it in many ways. And it's easy to work with, and we have the transgenic animals. So it's on the one hand complicated enough to ask questions about how morphogenesis of a multicellular animal happens, yet it's relatively simple. Okay, so this is a good point to stop for questions before I go into the first story on the nematic fiber organization in regenerating Hydra.
Are there any questions so far? If there are, please type them in the chat box. Yeah, Samarth, please go ahead.
Yes, hi. So in the first part of the talk, you showed a video of Hydra regeneration. So in that, I want to know what is the dark patch in the middle, which is then towards the end also in the video.
Go back.
In the middle of the body, there seems to be I think I saw a dark patch. I want to know what
it might be there. So during this process, there can be cells that die, and so basically that patch is within the cavity. It's probably cells that died following the regeneration and are eventually secreted from the mouth.
Okay. Okay.
So this is not part of the. I mean, the body itself is the body itself is the tubular surrounding, and the things in the cavity are, that's where it digests its food and it pushes back, then it secretes it. It's not part of the animal itself.
Okay, thank you.
So there's a question from Roni, and she has audio problems, so I will read it out. So here, Roni asks, how does one generate transgenics in Hydra?
So basically, what you do, as in many other species, you inject embryos with DNA, and it goes into the nucleus, and then you generate transgenic. It's not very well controlled. You don't know where it inserts, but you're able to generate these transgenics that so basically by genetic manipulation of the embryos, and then you grow them. One nice feature about Hydra is because they're able to regenerate. If you have only part of the cells that are labeled, you can cut that piece off and get a full transgenic animal, or you can combine transgenics by mixing cells from different animals through this regeneration process.
Maitri, you have a question?
Yeah, I was just trying to get a sense of how the two layers, ectoderm and endoderm, are arranged. Is it that the basal surface of the ectoderm is separated from the basal surface of the endoderm through this extracellular matrix?
Yes, exactly. So basically these cells are arranged so their apical layers are on the outer side in the ectoderm and on the gastric side on the endoderm, and then their basal layers are both sandwich the extracellular matrix in the middle.
Got it. Thank you.
Manjunath?
Hello. So can I go ahead?
Yes, please go ahead, Manjunath.
Yes. Thank you for your talk. So I was just wondering about the criticality hypothesis, or you're going to cover this a little later. So I'm aware of the paper criticality at morphogenesis in PNAS, which appeared few years ago. And also I'm aware of some literature on criticality where they say that before morphogenesis, the cells become so sensitive to the external stimuli that they actually, if I were to say, maximize their energy efficiency in dividing. Is that true, or is it like it's just only an hypothesis?
I cannot really comment on. I cannot really comment on this from our work in a concrete way.
No issues. Okay, then. Thank you.
Apratim Chatterjee, please go ahead.
So is there a minimal size beyond which they regenerate? I mean, if you have 2 or 3 cells, they wouldn't regenerate, right? I mean,
yeah. So there is definitely a minimal size, and there are experiments characterizing this. So both if you cut a tissue piece, if you cut a too small tissue piece, what happens is that it will round up into a spheroid, but it will disintegrate after a while. And similarly, when you generate cell aggregates, there's a minimal number of cells that is required for this. What sets this minimum is not clear, but definitely you see that there is a minimal size.
Okay, so I'm not seeing any more questions in the chat box, but Kinneret, I have a question. So
Yes.
These tentacles, is there a stereotypic number that come up, or is this completely random?
No, it's actually variable, and it definitely depends on the size of the animal. So typically, when you regenerate, and initially before the animal feeds, it's smaller. So then you'll have fewer tentacles, and it's not a well-defined number. There seems to be a kind of characteristic distance between tentacles.
Interesting. Okay. Okay, so there are no more questions at this time, so please go ahead.
Okay, so in the first part of my talk, I'll focus on the nematic fiber organization in regenerating Hydra. So as I showed you, I'll be focusing on the ectodermal fibers because they're easier to visualize. And if you look at the full, this is an image of a full small Hydra, and you see these actin fibers, and you can locally define their orientation. This is what's called nematic organization. So if you have elongated objects and they're aligning along a preferred direction, you can describe this direction by what's called the director field, but basically a headless vector that is locally aligned with the orientation. And before going to our work on Hydra and nematic organization in the actin cytoskeleton Hydra, I want to just give you a brief overview of nematics and active nematics to give you some background about this. So nematic organization has been known in a large number of material, maybe mostly studied in the context of what's called liquid crystals, which are molecules which are elongated. And what people found is that if you look at the phases of these molecules, you actually have an intermediate phase between the liquid phase, which is fully isotropic, and the crystalline solid phase, which is ordered. There is what's called a nematic phase in the middle, where on the one hand, it doesn't have the organization of the crystal, so it doesn't have a well-defined long-range organization. But on the other hand, it's orientationally organized. So it's lost its the rotational symmetry of the isotropic phase. You see that the molecules are aligned along a preferred direction. And this intermediate phase is called a nematic phase, characterized by this nematic organization. And you can see this type of organization in many systems, both in animate systems like these liquid crystals, and also in live systems. Here you can see an example from cytoskeletal networks, which I'll talk about a little more in a few slides, from Zvonimir's Dodik lab. So what they have here are microtubules and in motors that will make this active nematic system. And you can see the alignment of the microtubules in this nematic organization. Here you can see an example, a school of fish. And again, you can see that there's a alignment. Here, actually, this is polar order because the fish has a direction. And one important aspect of these nematic material is that there can also be defects in the nematic. So a nematic is characterized by this director field, which wants to be locally parallel, but there can be singularities in these fields. And if they have point singularities, these are what's called topological defects. And you can characterize these defects according to their structure. And this structure reflects the topological charge of the defect. So the topological charge of the defect is just the winding number. So if I go around the defect, for example, I have here a defect. If I go around this defect, then I can ask how much does the director field rotate as I go along the path around the defect. And in this case, this winding number is 1. I do a full circle. So this is a defect with a topological charge of 1. Here you can see a defect with a topological charge of half. If I go around here, I do only half a rotation. And this is minus half. In this case, I do half a rotation, but in the other direction. Now, an important feature of this these topological charges is that they're constrained. So if I look at the topology, for example, of a spheroid, if I have a closed spheroid, then the total charge on the spheroid is constrained by topology to be equal to 2. So you actually are familiar with this. If you think of a globe with longitudinal lines, you know that there have to be poles. They cannot be parallel everywhere. And each of these poles is a +1 defect, which gives you a total charge of +2. Now there's a class of nematics which are called active nematics. This is a subclass of what's called active matter. And these are out of equilibrium systems where the constituents of the system actually use energy to generate forces or self-propel themselves. And here you can see this example I mentioned earlier of microtubules that are connected with molecular motors. And you can see this nematic organization with the defects. And if I play the movie, you can see that this is actually an active system. These defects are able to move, and you have self-organized flows in this system. So this is a class of system that has been studied in the last decade or so, and shows very interesting behaviors. You can see spontaneous flows like you saw in the movie. You can see generation and annihilation of defects. So here you see an example of generation of defect. Initially you have no defect, and then it splits into a minus half and a plus half defect. These defects can be motile. So here you can see this plus half defect moving in the direction of its rounded end. This tells you about the stresses in the system. In this case, this is an extensile stress. You can also look at monolayers of cells. You can see here different types of cells, and these are all elongated cells. So these are bacterial cells or epithelial cells, which you can which have a long axis. And again, you can describe these as active nematic systems. These cells are able to generate forces and move about. And you can look at the organization and the defect dynamics in these systems. And the thing I want to highlight, and will be relevant for our work later, is that if you look at these systems, the topological defects actually tell you that at these sites, there will be a unique mechanical environment. So you can see here two different examples. You can see here examples from bacterial colonies, where you look at the bacterial colonies, and you can look at the flow pattern of the bacteria, and you can see that there's a specialized flow pattern around the defect. So here you can see this is the site of the defect, and you can see the flow pattern around that defect in experiments and in simulations. Here you can see an example of epithelial cells. And again, if you look at the region around the defects, here you can see what you're seeing here is the stress pattern around the defect. And you can again see that there's a unique mechanical environment at the defect sites. And this unique mechanical environment can generate various specialized phenomena at that defect sites. So again, showing you here examples from in vitro layers of cells. You can see that at the defect sites, you have cell extrusion, which leads to apoptosis and cell death. You can see here examples where you have an accumulation of cells. So cells, a cell mound forms at the defect sites. And here in this example of layers of bacteria, these layers actually form, you start with a single layer, but over time you develop multi-layer structures which start up at specific defects. So what I want you to take from this is that, okay, first the definition of a nematic and the definition of the topological defects, and then to understand that these can be active and there can be defects, and these defects are specialized sites which can lead to phenomena. And what I'll show you in our work in Hydra, and that these specialized sites can actually be relevant for the process of morphogenesis. So maybe I'll stop here briefly for questions on the nematics, and then go on to describe our work in Hydra.
Anyone has any questions so far? Yeah, Atiyab, please go ahead.
Yeah, can you hear me?
Yes.
Yeah, nice lecture, by the way. You know, I can't help but notice that my fingerprints also have this particular pattern, the nematic pattern. Is it so that you can probably look at these defects and identify the fingerprints from one person and the other person?
That's exactly definitely true. And in fact, the software that we use to analyze our images of Hydra is exactly software developed for fingerprint analysis. And it's definitely true that the types of defect and the pattern or defects are an important part of what people use for fingerprint analysis. It's certainly true.
Interesting, very interesting. Thanks.
Maitreyi?
Yeah, so you showed us three kinds of effects surrounding nematic defect, so extrusion, mound formation, and stratification. What determines how a sheet will respond to a nematic defect?
Okay, so without going into too much detail, first the type of defect is important. So these, for example, in the examples I showed you, the plus half defects have different behavior from the minus half defect because of their different structures. And then in each system you will have in each system that you have the entities that generate the forces or self-propel or whatever process they do. So that will determine the types of forces within this sheet. So in general, in active nematic, you will talk about if you have a situation where you have contractile forces or extensile forces, and these will change what the pattern around the defect will. So the things that matter are what types of forces are generated by the entities within the monolayer, and what type of defect which controls the structure. For example, one very important feature of the defect is the symmetry around the defect. So if you have a symmetric or non-symmetric organization, that can have a large influence.
Thank you.
I don't see any further questions, Kinneret.
Okay, so that was more an introduction, and now I want to jump into our work on the nematic organization in Hydra. So as I just mentioned in answering the question, we are computationally extracting these director fields from the image intensity gradients using this software developed for fingerprint analysis. This organization of the actin fibers is very stable, and just to illustrate that, you can see here an example where you're looking at the cell division. So within this epithelial layer, you can have cell divisions. You can see here a cell, and you can see that it's dividing into two cells. And what you can nicely see here is that the actin fiber organization remains stable throughout this process. So this nematic actin orientation of these supracellular actin fibers is a very stable feature in this system, both in space and in time. In space, this is reflected by the relatively low curvature on cellular scales. Okay, so if we look at the mature Hydra, we can see that there are defects, and we can see that these defects are not distributed randomly in the animal, but they actually coincide with the main morphological feature of the animal. So here you see an image of a Hydra. You can see that we have a +1 defect at the tip of the mouth, a +1 defect at the tip of the foot, and each of the tentacles has a +1 defect at its tip and 2 -1/2 defects at its base. So altogether, this +1 defect at the head and the foot give us the +2 charge, sorry, which is required by topology for something that has the topology of a spheroid. And then each of the tentacles actually doesn't contribute to the total charge because the +1 defect at the tip is balanced by the 2 -1/2 defects at the base. So the total charge of a tentacle is actually 0. And then you can have a variable number of tentacles that do not contribute to the total charge. There are cases where we see multi-axis Hydra. So here you can see a scheme and an image of a 2-headed Hydra. And then this extra +1 defect at the extra head in this case will be balanced by 2 -1/2 defects at the junction of the animal. So this is the pattern of defects. If you look at the full Hydra, this was in the ectoderm layer. We see a similar pattern of defects if we look at the endoderm layer. So again, we have a +1 defect at the mouth and a +1 defect at the foot, at the tip of the tentacle, and -1/2 defects at the base. You can see here that because the fibers in the ectoderm and the ectoderm are perpendicular to each other, the type of +1 defect here is not it's a vortex rather than the aster that we saw before, but we see the same pattern of defects. Okay, so now let's go into the process of regeneration. So I told you that we can cut a piece of the animal, and this small piece will regenerate into a full what we saw is that when you cut out the piece, so let's say we take the tubular body and we cut out a square fragment from this tubular body, you see that this piece inherits the actin fibers from the Hydra. And what we were able to show in this paper a few years ago is that this inherited actin organization actually provides a structural memory of the alignment of the body axis. However, this cannot happen trivially, and because this square piece of tissue first has to fold into a sphere. And when you fold into a sphere, you cannot maintain your nematic organization everywhere. What we see, in fact, is when this piece of tissue folds into a sphere, you can see here images. What we see is that the tissue folds in such a way that in the central part of the fragment, when it falls into a spheroid, the fibers are actually preserved, but in the closure regions, the fibers are essentially lost. So what you have is that this open square closes into a spheroid in a purse string-like manner. In the closure regions, the fibers are lost, and in the central regions, the fibers remain. So we have here a partially ordered array of parallel actin fibers without defects, but with this large disorganized region. What we see is that during the first 24 hours of the regeneration, after the sphere folds, it starts regenerating. What we see is the process of induction of order. So what we see is that over time, disordered regions extend, and this disordered regions goes away. And you can see this here in this scheme. What we've done here is quantified the region that is ordered from the fraction of area of the spheroid that is ordered. And you can see that it starts at about half and then increases over time. And at about 24 hours, it reaches a stable level close to 1. So over time, what we have is an induction of order. So these regions, these regions that are ordered, the disorder invades the disordered region. And what we have is a spheroid that has a full array of ordered fibers. Now, I told you before, if we have a spheroid with a nematic array and it's ordered, there have to be defects. These are the type of defects that we see in regenerating Hydra. We see plus half defects. So what you're seeing here, and you'll be seeing in my further slides, is here you're seeing the image of the actin fibers labeled with LifeAct GFP. On the bottom here, you're seeing what's called the local order parameter. So these black lines give you the local direction of the actin fibers, and the local order parameter is a measure of how locally ordered these are. In regions where the fibers are parallel, this will be close to 1. In regions close to a defect, this will give you a minima. So you can clearly see the defects as minima in the local order parameter. And you can see here a plus half defect, a minus half defect, and a plus 1 defect. Now, if we think of the process of morphogenesis, so we start with this defect-free square fragment. It folds, you have induction of order, which means that you have a spheroid that has an organization of actin and must contain defects. These actin organization is rather stable, so these defects are long-lived. And eventually this has to regenerate into an animal with a stereotypical defect configuration that I showed you with the plus 1 defects at the head and the foot. And the question we were looking at is if we can, by looking at this nematic organization, if you can develop an effective description of the morphogenesis process, and if that is able to give us some predictive power over how this how and where this these morphological features will appear. So to do that, we wanted to follow the actin dynamics in regenerating Hydra. We want to see the process from all directions, so we went to light sheet microscopy. So what you're seeing here is a regenerating spheroid, where we were looking, each of these images is from a 90-degree rotation, so you're seeing it from 4 different angles. This is the initial folded spheroid. You see the ordered region in this side. You see this region is disordered. So on the top, you see the actin fibers. On the bottom, you see the ordered parameters. The shaded regions are the disordered regions, and you see the local order parameter. And what you can see is how over time, you have stable defects that are forming, and these defects are dynamic in the tissue, and eventually this will lead to the formation of the characteristic animal. So now you can already see that the head is developing on this side. You see the tentacles emerging, the +1 defect at the tip of the mouth, the +1 defect at the foot. And over time, you see how this nematic field evolves, and the Hydra regenerates into a small animal. So at the end, we see the mouth with a +1 defect, the tentacles, and the foot. So let's look at the evolution of the nematic field more closely. So as I told you, we see +half defects, -half defects, and +1 defects. What you're seeing here is quantification of the number of defects from each type as a function of time over the regeneration process. During the first 24 hours, we have regions that are still disordered, and by 24 hours, we have all the disordered regions have disappeared, and the defects form from this disordered region. So initially, we have no defects in an open boundary. We have this disordered regions. You see that the most prominent defect that appears early on is the +half defect. However, this defect is entirely absent from the full animal, and you see it disappearing over time. So by 3 days, when we have a full animal, these +half defects disappear. And what we see is the emergence of the +1 defect at the head and the mouth. I should note that in this scheme, we did not include the tentacles, so the defects associated with the tentacles, because the tentacles grow later in the movie. So we focus on the defects that set the body axis. You can see that the total charge of the defect stabilizes at 2, as it must from the constraints of topology, and stays that way. During this time, however, the configuration of the defects evolved from having mostly +half defects to having 2 +1 defects. During the initial stage, the net charge is smaller than half because of the large disordered regions. So how does this, let's look more closely on the dynamics and the evolution of the nematic field. So one thing we notice is that if you look at the +half defects, these +half defects are actually motile. So you can see this here. To look at the motion of defects relative to the underlying cell layer, we used a landmark. We introduced a caged fluorescent dye and uncaged it in a particular region. So this allows us to label specific cells. So these labeled cells are shown here in green. You can see a +half defect here, and you can see over time that this +half defects moves away from these labeled cells. So the +half defects are mobile, and I showed you that they disappear. How do they disappear? The most common way they disappear is merging of 2 +half defects. So what we see is that there's 2 +half defects facing each other, and eventually these will merge. And as I'll show you, this will typically become the foot of the animal. What we also see is that the +half defect can also annihilate with a -half defect. So this is what you can see here. You have a +half defect here, a -half defect here, and over time they merge with each other and disappear. What was very striking is that we saw that the formation of the head and foot is directly related to the dynamics of the defect. So when we have 2 +half defects that merge, if we look at the outcome of this +1 defect, we see that this will very likely become the foot of the animal. So what you're seeing here is the outcome of this of the merging of 2 defects, and you can see that this will become the foot of the regenerated animal. Contrary to that, we see that if we look at +1 defects, these actually remain stationary relative to the labeled cells. So here you can see a +1 defect early on, and you can see that it remains in the region of the labeled cells in the regenerated animal. And these +1 defects that we see early will develop into the head of the animal. So what this means is that by looking at the the nematic organization of the actin fibers early on in the spheroid, we can predict to a very good approximation where the morphological features of the animal will appear much before we see any sign of this. So we can say that this region will develop into the foot and this region will develop into the head much before we see any sign of this in the morphology of the animal. So this brings the very interesting question of how are these topological defects actually related to the formation of functional features. So I showed you that we can label this region, and we know that this +1 defect, early +1 defect, will develop into the mouth of the animal. But obviously the mouth of the animal has to develop a mouth, you have to undergo many biological changes. It's not just the actin organization. You have to have cells that differentiate and have specific functions at this mouth. So what's special about these +1 defect? So as I alluded to earlier in my introduction on active nematic, the mechanical environment at this site is special. In this case, what we have here is an aster of contractile fibers. These contractile fibers mediate the interaction between cells. So at the core of this defect, we expect there to be weaker adhesion because there are no fibers spanning the cell-cell contacts in the core of this defect. And also, when all these fibers, which are muscle fibers, they contract, we expect this region to have a high tension. So the mechanical environment in this region will be characterized by weaker adhesion and higher tension when these fibers contract. And this is actually nicely reflected in an observation that we made. If we look at the Hydra, Hydra is you saw that it's a tubular structure filled with fluid, and there's actually an osmotic influx of material that leads to osmotic inflation of the tissue. And when you look at the dynamics of the tissue, you see these rounds of inflation and then rupture, inflation and rupture due to this osmotic effect. And when we looked where these ruptures appear, we see that these ruptures happen specifically at these sites of +1 defect, as you can see here. So you can see here, this is a +1 defect here. You can see the hole forming here and then closing. And the mechanical environment in these +1 defect with this weaker adhesion and higher tension makes them susceptible to this formation of rupture hole. And here you can just see the quantification of where the holes form. We never see holes form in ordered regions. And once these +1 defects appear from the disordered regions, we see that the holes predominantly form in these +1 defects. Okay, so I've shown you that the +1 defects appear early on and remain stable and become the head of the mouth of the animal. I've shown you that rupture holes form at these +1 defect sites. From their structure, we know that the mechanical environment at these +1 defect sites is special, and then they preferably become the head of the animal. So what we want to say from all these observations is that the +1 defects effectively act as organization centers for the process of head formation. They're there early on, they're stable, they remain in the same place, and these cells in this place experience this unique mechanical environment and eventually regenerate to form the head. So to summarize this first part, I've shown you that active nematics provide a useful coarse-grained framework for looking at the dynamics of the morphogenesis process and the actin fiber organization in regenerating Hydra. And I've shown you that this gives us some predictive ability. We can identify these +1 defect sites early, they remain stable, and they eventually become the morphological features of the animal. So these +1 defect sites effectively act as the organization centers for the formation of a head and a foot. And as I said, this has recently been published. So this is a good place to take some questions before I move to the second part on polarity.
Thanks, Kinneret. So we have Maitreyi.
I was just trying to understand. So you cut out a sort of squarish piece of tissue that's bilayered, and then it becomes spherical. So is this location of this +1 defect where the sort of wound closure type of thing happens? Meaning that is that where you're
yes,
sort of circling the square, so to speak?
Yes, but it's so Yes, but this wound is quite broad. So basically we have this if you look at the initial spheroid, then about half of it has this nematic array without any defect, and this will remain organized. The defects will appear in this disordered region as you close the system, and within this region, this will become the mass. So yes, it forms in this disordered region, but I just want to emphasize this is not it's not as if it forms at that it closes into a point where that becomes the head.
But so if you look at if you label the cell membranes, do you have good cell-cell contacts in that region? I mean, so
yes,
are the contacts there different from elsewhere? You said weak adhesion, but I was just trying to ask whether the morphology of the contacts in those regions is actually different.
Okay, so at when the tissue closes, since you see this osmotic, osmotic inflations, etc., the tissue is overall, it's the adhesions are connected well. Locally at this region, yes, I anticipate there to be some difference, but we're not in a, you saw the movie, it's very hard to track individual cells within this dramatic movement. So we're not at the ability to follow individual cells, but overall cells are connected. But yes, I would say that the mechanical environment in this +1 defect site is different, and that is leading to the behavior we see. But I cannot at the level, like at the molecular level of what's happening there, no.
Right. Okay, thank you.
Senthil?
Hi, Kinneret. Wonderful talk and nice analysis. I just want a clarification regarding, are you saying that the nematic organization is the defining feature of where the mouth forms, or are there no underlying other biochemical interactions that may determine this?
Definitely not, and you'll see in the next section when I talk about polarity. Definitely not. So I think it's an interplay of these biochemical signals and this mechanics. What I'm saying is that the mechanics is not a passive counterpart, but actually that the local, the nematic organization and the mechanical environment that it ensues influences the process, but definitely not without relation to the biochemical signaling. And the second part of my talk where I focus on polarity exactly touches that and shows this interplay.
Mm-hmm. And I was just wondering if you tried experiments where you can use, for example, an infrared laser to create local defects or some actin optogenetic modulation to drive the defects and see if it reorganizes globally?
Mm-hmm. So one thing to appreciate is that actually by locally perturbing something, you cannot generate a localized defect. And the reason is that you have much conservation. So if you, for example, locally disrupt the actin fibers in a region, what will happen is that you might have transiently, you might have some defects. They'll have to have a net charge of 0, and they will very quickly reorganize into not having a defect. So in order to induce a defect, you have to work harder than that. And that's definitely something we're interested in working on to try to do it. But you just have to bear in mind that you're not able to generate, for example, a +1 defect on its own, because the region had, if you're not changing the boundaries, the region had a net charge of 0.
All right, thank you very much.
Sandhya?
She said she was going to address the question. Essentially, my question was, if both head and foot have this +1 defect, how do you decide whether you make a head or a foot? And I guess this part of the talk is going to address that.
Yes, so I'll just say in the context of the fragments that we looked on here, what we see is that eventually both the head and the foot will have a +1 defect, but we definitely see a difference in the dynamics of how these defects come about. So we see that the +1 defect arises earlier, whereas for the head, and the +1 defect that becomes the foot actually typically comes from merging of 2 +½ defects that will merge very late in the process. So we definitely see a difference in the organization, even though eventually both will have a +1 defect. And yes, as you said, I will address in the second part of the talk this relationship. And I want to emphasize it's not a causal relation. It's not that the +1 defect will become a head. Obviously, that is not true because we have +1 defects at the tip of the tentacles and at the foot. And you can also, as I'll show you, can also generate a +1 defect. So it's neither required nor sufficient, yet it's influential on this process.
Thank you.
Okay, Kinneret, I have a couple of questions.
Okay,
so what is the mechanism for the ordering of the actin fibers initially?
Mm-hmm.
Do you know?
So that's a good question. I mean, we don't have a good answer for this, but I can say that it looks similar to what people have looked at in vitro in cells and in other systems, where basically the idea is that mechanical coupling through the matrix induces this nematic order. So in other systems, for example, in just adherent cells, what people saw is that if you take the matrix and you start pulling on it in a oriented fashion, then you can get ordering of the fibers within cells. So likely this is a process that involves mechanical feedback, where in our case, the actin fibers themselves generate the main generators of the forces within the tissues, and then cells respond to these forces and align the fibers, and this is mediated by coupling through the matrix.
Mm-hmm. But then here the fibers span cells, or how does that work? So it looks like here the fibers are much longer than individual cells themselves, right?
Yes. Yes. So
these are extracellular actin, or how does
okay, so first there's no extracellular actin. Actin is always inside the cells. What you have in this case is indeed that, from as you see in my images, mechanically these actin fibers actually appear to be connected across the whole organism. What you have is basically that in each cell you have these actin fibers, and they actually these actin fibers actually protrude through filopodia-like structures in both regions, and then you have overlap between fibers from adjacent cells, but all this is connected. So basically you have the fibers underlying each cell and then protruding, and then they're connected through cell junctions to other fibers. So you have a mechanical connection between actin fibers from neighboring cells, which leads to this supracellular structure.
Wow, wow. So there's a lot of coordination.
Yes.
Right. And so my last question is maybe a slightly technical one. So in one of the slides, you quantified the number of defects. And is it right that I noticed that there was a fractional number of defects?
Yeah, yes, this is
something there.
So what we did here, is just look at the population of samples at different time points, and we measure for each sample how many defects of each type we see. And this varies between samples. What you're seeing here is just the average, and the shaded regions is the standard deviation. So these are fractional, it's not fractional defects, but it's just an average number.
Okay, okay. Thank you. And right now I don't see any more questions, so please proceed to the next part.
Okay, so in the next part, I want to address this question of polarity, which is also related to the question I was asked regarding how this relates to biochemical signaling. So on the one hand, I showed you this structural memory of the body axis alignment based on looking at the actin fibers. On the other hand, it's well known in Hydra that there's a memory of polarity, and this has been known for a long while. So from the original works on Hydra from Tremblay in the 18th century, if you cut a Hydra into two, then you see that the polarity is maintained. So this head will grow a foot and the foot will grow a head. So the polarity of the original animal is maintained. And it's also been known that if you cut a ring of Hydra, so you get rid of the head, you get rid of the foot, you just have a ring, and you look at how it regenerates, it will remember its polarity. So the head side will grow a head and the foot side will grow a foot. And here you can see examples from our data. What we do here is just to maintain the polarity of the animal, we thread it on a wire and it can regenerate on the wire, and you can see that the head side grows the head and the foot side grows the foot. So there's obviously a memory of polarity, and there's quite a extensive literature on the signaling that is specialized at the head and the head organizer in the animal that's related to this memory of polarity. What we wanted to see is what happens when we try to perturb the system, to understand how this polarity, the polarity of the animal is defined. We wanted to perturb the system and look how it reacts, and learn from this on this. What are the processes important for establishing polarity? And what we did was we actually made, we took rings from two samples and we placed them together in frustrating conditions. So here you can see what we call a head-to-head configuration. So what do? We take rings from two animals. Each of these rings has a head side and a foot side. But what we did was we fused the head sides to each other. So what we have here is a configuration where we have what we call a ring doublet. So we have two rings and their head sides are facing each other. And so we have frustration in the sense that these head sides are not at the edge of the individual ring, but they're actually in the middle. And what we sought to do here is to look at these, how the animal reorganizes from these frustrating conditions and try to learn from this on what's important for establishing and maintaining body axis polarity. So this is how these fused ring doublets look like. So we label each of these rings in a different fluorescent color so we can discriminate between them. And when we look at these head-to-head doublets and we allow them to regenerate, so the majority of animals regenerate, indeed a large fraction of them develops a head in the middle, which is kind of what you'd expect if there's a strong incentive to make a head on the head side. And this is how this looks. So you can see here, you see half of it is labeled with magenta, half is labeled in green. You see the dynamics of regeneration, and you see a bump starting to form at the middle, and this will develop into a head in the middle, and the two sides will develop into feet. But what we also saw that happens is quite striking. So if you think of it structurally, this looks like a single ring that closes at the top and in bottom. And what we saw is in a significant fraction of the samples, what we see is that head formation is actually suppressed at the middle. And what we see is that the head actually forms at the foot edge. So in this case, again, it's a head-to-head configuration. Both of the edges are originally at the foot side. And what you can see is that actually in this case, we have a head developing into from a originally foot-facing side. So in this case, we have what we call polarity reversal. Basically, we have originally, if you would take this individual ring, this part will always regrow a foot, and this part will always regrow a head. When we put these ring doublets together, we see in this case suppression of head formation in the middle, and we see a head formation at the end. And what's most striking here is that it doesn't form, it forms actually in the region that was furthest away from the head in the original animal. We can also look at the influence of introducing some perturbation into this head-to-head interface. So what we see is that a head which would want to form in the middle, when we put the two pieces of tissue adjacent to each other, is often suppressed. But it seems that the second tissue is somehow interrupting the formation of the head. If we put some type of physical barrier between them, we see that this is largely removed. So in this case, we see that nearly all the animals form a head. So by having a region where there's no adjacent tissue, somehow this promotes having a head in the middle. Another thing you can do with these ring doublets is that we can play with the configuration of the doublets, both the orientation and also where the tissue comes from. And we can look at how the original position of the tissue influences this. Now, when we do these experiments in the head-to-head configuration, we don't see much difference because the most dominant thing there is the frustration of polarity. But what we can do is we can look at the head-to-foot configuration. So in this case, what we do is we take these two rings and we put them one on top the other in the same orientation. So the head side of one goes to the foot side of the other. But we can do this head-to-foot ring doublets in two ways. We can put the tissue in the order that it was in the original animal. So we can label the ring that is above the midline by U and the ring that is below the midline by L. And I can take the upper ring and put it on top of the lower ring like it was in the original animal. Or I can take the lower ring and put it on top of the upper ring. So in this case, the orientation of the two rings is the same, but I have a region from the bottom part of the body that is on top of the region that is on top of the this. In both cases, both rings have the same orientation. So in this case, the doublet has a preferred orientation. What we see is that in the oriented configuration, basically the regeneration happens perfectly nearly all the time. So we see a head form on the head side and a foot form on the foot side, and the suppression of a head at the interface, and we get nearly all the animals regenerate into this normal morphology. However, when we switch the organization of the tissue, so we have the lower part above the upper part, we see that this is very, very different. So this emphasizes the importance of the where the tissue comes from along the original body axis. So we see a dramatic effect if we depending on where we took the rings. And what's most dramatic here is that in some cases we get again polarity reversal, so a head growing on the foot side. But in this case, actually, if you think about it, both rings switch polarity. So in this case, what we have is we put the lower ring above the upper ring, and what we have is that the head emerges from the upper ring, from the foot side of it. Both rings switch polarity, so it doesn't grow along the original polarity; it goes in the opposite polarity, again with a head forming on the originally foot-facing sides. So this is quite striking in the sense that the memory of polarity is really flipped in this tissue. What we also see in these experiments is that we don't see many instances of two heads forming. In most cases, we have one head forming, which suggests that the formation of a first head suppresses the formation of a second head. To look at that process specifically, what we did was we made ring doublets in what we call the foot-to-foot configuration, where we have a free head facing side at both edges of the ring doublet. So in this case, we put in the two rings, their foot side adjacent to each other, and we have a free end that can generate a head on either side. And we look to see if we regenerate into two heads or we regenerate into a single head. And what we see is that indeed we see suppression of two heads. We see some two-headed animals, as you can see here, but most of the animals regenerate only one head. And what was also very striking is that if you looked at where this head appears always on the upper side. So again, we see this memory of where the tissue was in the original animal reflected in the regeneration processes. So the head side on the U is more likely to, is actually always the one that will regenerate the single head in cases that we see a single head. So if I put all these experiments together, we see that whereas in the initial animal, we have this perfect memory of polarity. We have the head side always turning into a head and the foot side always turning into a foot. When we put these two rings together, we see that the structure actually, both the structure and where the tissue originally came from matter for what happens. So you can see the importance of the structure in the sense that the head formation is preferable at the edge. So when we take these ring doublets, we see that at the edge, we can have head formation even on the foot side. And again, if you think of the, in terms of the signaling, the common picture is that you have something, something at the head that marks the head side, and then you're least likely to form a head on the regions that's furthest from the head. But this is exactly what we see in these cases. And you can also see the importance of the structure in the suppression of the head in the middle. So when you put another tissue adjacent to the tissue, that suppresses the formation of the head, even in the head-to-head cases where you have a high bias to form a head, if you think about the original polarity. So now going back to relating this to what I showed you in the earlier part of my talk related to the actin structure. So when we take this, when you take a single ring, this single ring has this array of actin fiber, and then when it closes, it will form a +1 defect at the tip of the at both sides, both at the what will become the head side and what will become the foot side. And as I showed you in individual ring, this polarity is perfectly maintained, and you can see this in these images here. So in this case, we label the +1, the region that was close to the head side defect that will quickly close and form a +1 defect at the originally head side. And you see that this +1 defect will become the head of the regenerated animal. Now let's go to a ring doublet experiment. So here we take our two rings, each of them from a different tissue, and we put them together. And now structurally, after this, structure heals, what we have is something that looks like one thick ring. So we have a +1 defect at the one edge and a +1 defect at the other edge, whereas the region between these is defect-free. You can see here, this is tissue coming from one ring, tissue coming from the other ring. You can see the +1 defect at the edge. You can see, you can't actually see this because it's very smoothly connected between the two regions and the +1 defect at the edge. So now let's look at what happens in the polarity reversal. So again, we're going to the head-to-head configuration. We have our two rings with their head facing towards the middle. They form this ring doublet that structurally has a +1 defect at the both ends, both edges. And what we see is that in this case, I showed you earlier, sometimes the head will form in the middle, but in cases of polarity reversal, we see that the head doesn't form randomly anywhere in the body, but it actually forms specifically at the +1 defect side that forms very early. So what you're seeing here, these are all images from the same movie, from the same time-lapse movie. And you can see here very early on, we have a +1 defect at the head. You can see here, this is from a side view, this is from a top view. This allows you to see the defect nicely from the top view. So you see that this defect forms very early on. And when a head eventually forms, it will form exactly at this site. So, and from a kind of a signaling perspective or from proximity, the memory of polarity, this is the least likely sites for head formation because this was the site that was furthest away from the head. So in this sense, the +1 defect size seems to act as an attractor for head formation. The head doesn't form randomly, it forms specifically at the +1 defect site. In other cases, as I showed you earlier, we can have the head forms in the middle. And in this case, this will involve defect formation, new defect formation, like you see in budding of Hydra, for example, where a new head emerges in the middle. So I think this nicely demonstrates the relations, the interplay between the signaling and mechanics and cytoskeletal organization in this process of regeneration. So we have this ring doublet, which is under frustrating condition. Signaling wise, it would want to make its head in the middle. The structure has the defects already ready at the edges. What we see is that sometimes we observe polarity reversal where the new head will form exactly at the +1 defect site at the edge. Whereas in other cases, the signals that ignite this, the formation of a head in the middle win, and we get a head in the middle. So this really nicely shows this interplay. A defect is not required for head formation. We can have a defect form without a head form without a pre-existing defect. But however, a pre-existing defect can act as an attractor for head formation. Okay, so to summarize this second part, I've shown you this these experiments with these ring doublets, which allows us to look at reorganization of polarity in a kind of a clean way where we can generate frustration in a controlled manner. And by looking at these, the regeneration of these ring doublets, we can see that even though body axis polarity is very, there's a strong memory of this body axis polarity in Hydra tissue. When we put it under constraints, it actually shows that it's actually dynamic. It can change and even reverse. I've shown you that this that the polarity depends on the memory of polarity in the original tissue, which comes from its direction in the original body, as well as the position of this the position of the ring along the body axis of the animal. But I've also shown you that the +1 defect or the structure can also matter here. And we see that in the examples where we put the system under frustration, we see that the +1 defects can actually act as attractors for head formation, very much in line with what I showed you earlier. So even though a +1 defect is not required or sufficient for head formation, the specialized mechanical environment at these sites promotes head formation. So to kind of wrap up everything together, I've shown you that if we look at the nematic organization of these supracellular actin fibers, we have a coarse-grained field that we can look at during the regeneration process. I've shown you that by looking at this this field without knowing anything else about all the other important processes that that are happening during Hydra regeneration, we actually, just from looking at the organization of the actin fibers, we can get an effective description of this process, and it's predictive in the sense that we can know that this region of this region that is characterized by these types of defects will lead to the formation of the head or the foot of the animal. And I've shown you how this, how the interplay between the biochemical signals, which are involved in this memory of polarity, with this organization of the nematic field, together lead to the regeneration of the body axis in these doublet experiments. So I just want to kind of highlight the picture that comes from these results. So we have this slowly varying field, which is the actin fiber orientation, which we can follow. This field will influence the environment that cells see, and specifically at the regions of defects, there will be a unique mechanical environment that is associated with with having a defect in this nematic organization. So we can think of this nematic orientation field as a mechanical morphogen that provides local information to cells and has its dynamics. It's not by its own, so it definitely interacts with other fields, but this basically allows us to think of the actin organization as the mechanic as an analog to the chemical morphogens that we know from the Turing pattern, and the kind of global features of these nematic actin fiber organization and the global constraints on it gives us some large-scale ability to organize things over a large scale, which can contribute to the robust formation of functional tissues in Hydra. And with this, I'd like to conclude and thank my coworkers on this project. So this is a joint work with the lab of Erez Brown, who will give the talk in the afternoon. What I've shown you in the first part was regarding the nematic organization of actin fibers, was largely the work of Yonit Marudis-Dax, who's a graduate student in my lab, and the second part on polarity is work by Anton and Diora. And also, if anyone's I'm happy to take questions, and if anyone is interested to work on this fascinating organism, please also contact me for postdoctoral graduate positions. So thanks, and I'm happy to take more questions.
Thank you so much, Kinneret. That was a beautiful talk and beautifully organized as well. So for questions, first we have Raj Kumar. I think he got the answer to his question. So Roni, please go ahead. Roni, I can ask for you. Yes. Roni is asking, has your lab looked at any signaling pathways leading to suppression of head formation?
Yeah, that's definitely a direction that is really interesting to look at how this, so the main pathway that's associated with polarity in Hydra is the Wnt pathway. And if you look at a mature animal, it will have the organizer, which expresses Wnt at the head, at the +1 defect site. And looking at how the interplay between the signaling and the actin cytoskeleton is a really interesting direction. Unfortunately, there are no good live probes for looking at the Wnt distribution. So there are some transgenic animals that express GFP and a Wnt promoter, but these give a very late signal. So basically we see a signal arising only after we see a head and a tentacle. So there are no good live probes for looking dynamically at these signals, but certainly we can look at them just by fixing at different time points. What somewhat complicates this is that there's quite a large range of time dynamics. So it's hard to, without having a live probe, to correlate these. But it's definitely an interesting direction.
And Ronia has another question. Do polarity switch doublets take longer to form a normal body compared to a normal regeneration?
So that's actually also a great question. And I would want to say that I would perhaps expect that to be the case, but we have such high variability. Even if you look at the single-ring regeneration, there's quite a range of timescales that I think it's probably masked by the variation. So we don't see very striking differences in time, even though there could have been, but I think it's just that the spread of time is very large in these animals. There's quite a bit of variation.
And there's a third question also from Roni. Do Hydra have not signaling as there is contribution from contact between the doublets?
I'm not familiar with that, but I don't know.
Okay. Lakshya Chauhan.
Hi, this was a really good talk. I mean, I really enjoyed it. So I just wanted to know, like, could the reversal be seen real time? Like, if we use some fluorescent marker for proteins like HEDI and HYM301, which are known to be expressed only in either the tail or the head. So since some of your specimens showed a reversal of head and foot, could they be used, or have you thought of using them maybe to see the real time reversal?
I mean, that's definitely, we're really interested to see in seeing how this relates to kind of signals that are the earliest signal that is known to be associated with head formation is Wnt3 expression, and we have a Hydra that expresses that you can't visualize the Wnt itself, but it has a Wnt promoter that is labeling GFP. And all I can say is that to get measurable, like you see something after that's definitely a head just from the morphology. So that's not very helpful to seeing the dynamics. I mean, if anyone is able to generate strains that will give us indications of what's happening in terms of signaling early on, that will be really interesting, because that will allow us really to look at the dynamics of how between signaling and mechanics happens live. Currently, we don't have any live probes that are able to give us that information.
I see. Thank you.
Vijay, you have a question?
Yeah. So Kinneret, hi, fantastic talk as usual. So I just wanted to ask, in the doublet reversal things that you showed, is there a size limitation there? For example, rather than taking half and half, let's say you took 1/4 of one kind and the 3/4 of the other. Is there a bias that you would find?
That's an interesting question. So typically we take two rings that are about, something between 1/8 and 1/4 of the whole animal, and certainly I can imagine that there can be some a size dependence in this. It's yeah, but it's a bit tricky to look carefully because the experiment itself is quite like the grafting is quite difficult, and there's quite a bit of variation. So it's hard to say very carefully if there are some size dependence. And I would also say that all the sizes that we're looking at are kind of less than the typical size scale of a individual animal. So all of these are kind of, if you think, signaling wise, they're all within the size that a head will repress formation of other heads. Have looked at, large aggregates or like arrays when you put many tubes on larger scales, and then you can pass the kind of typical scale of an individual. But in all our experiments, we're all kind of within a smaller scale.
Mm-hmm. I was just wondering, for example, the Nematic organization would give some correlation length. And if that's the length on which there will be large-scale influences. For example, Planaria people have studied gluing things backside, and there's some chemical signal ingredients which control that. So I was wondering for the mechanical morphogen concept.
So again, also from a mechanical morphogen point of view, the kind of what we see in our system that you kind of it's a small system, so it's not generating many defects. Basically, you have kind of the minimal amount of defects to this again telling us that it's a small system also in that sense. So whatever correlation length you have for the actin are typically, I would say, larger than the size of the animal, but you're constrained to have defects because of the topology, so you have defects.
Vijay, does that answer your question?
Yeah.
Okay. Javier?
Yes, thank you. Thank you for this nice talk. So I remember some time ago reading about some works that studied the effect of thermal gradients during Hydra regeneration. So in order to determine actually the symmetry breaking and the elongation axis. So I wonder if you have considered studying how the nematic dynamics would actually be modified or changed due to these thermal gradients. Thank you.
Okay, so when you look at regeneration from excised tissue, actually our work shows that there is no symmetry breaking. Basically, that kind of the system comes in the actin organization is already set. I think that small changes in temperature also, will not affect the actin. These actin structures, once formed, they're rather stable. So I don't think small temperature effects like those reported in this paper will have any influence on the actin, the kind of inherited actin fiber organization. It is interesting if it would be possible to mess up the actin organization. That would be an interesting thing. The kind of the place where we already can see that was when you look at regeneration from aggregates, where initially you don't have this organization. But I don't think the kind of simple temperature picture of a nematic that is controlled by temperature from liquid crystals, say, is the same here. I think the system within the range that is viable, the system tends to be nematic.
Okay, thank you.
Sandhya?
That was really elegant experiments and beautiful data and wonderful talk. I had, I guess, a lot of my questions were answered. I was wondering, in the case where you made the head in the middle, can you induce, and you had 2 feet already, can you induce another +1 defect and get a 3rd foot? In effect, I'm asking 2 things. One is the presence of a +1 defect permissive for making either a head or a foot? And once you've made a head, every other +1 defect has to become a foot.
Okay, so definitely, the +1 defects are permissive for either head formation or foot formation. I think the most dominant process in Hydra regeneration is the formation of the what's called the head organizer, and that has been shown to generate signals that will repress the formation of additional heads, and definitely that's what we see. So I think definitely the notion that if you have a head, that will, to some extent, we sometimes see two-headed animals, but that's actually relatively rare in our data. So definitely the formation of a single head has a repressive effect. If that always means that the remaining +1 defects will become a foot, I'm not sure. Also in our experiments, we don't look directly at foot formation. I mean, we do see if the foot is also differentiated and has adhesive material that allows it to stick. In some of our experiments, we indeed see the functionality of the foot. It sticks to the substrate, but not in all experiments. So I don't know to answer directly if all the remaining +1 defects after we've suppressed further head formation do become foot, but quite likely I would say that would be the case.
Thank you.
Okay, so at this point, I'm not seeing any more questions, Kinneret. Thank you so much. That was a really beautiful talk. I enjoyed it very much.